Welcome, welcome, welcome once again to my channel. My name is Dr. Nchebe-jah, and today I want to make a very brief video for you on cell biology recap. It's extremely important. That's what you study in biology. The most important thing, all right? Now think about it. What is a cell? A cell is the structural and functional unit of a living organism. I want us to discuss this cell in different compartments, all right? At the end of this discourse, I want to give at least 10 to 20 questions. And by the ending, I'll give another 10 or 20 questions that you are most likely going to see in your exam. This is just a recap. Mind you, it doesn't substitute for your class and for your texts. So please, this is just a recap, something you look at few minutes or at the end or before your exam as a preparatory phase, all right? Extremely important. Now, I told you something in my prior videos that anatomy is what? Location, location, location. And physiology is what? Function, function, function. Another definition for anatomy is what? Structure, structure, structure. Extremely important. Now, why I mention this is because everything, both the structure and the function, is talking about cell, all right? I want to give a thorough recap of cell before I even discuss this PowerPoint. Cells are responsible for all structural and functional properties that an organism has, all right? Three major aspects where cell affects a complete organism. One is the workings of the human body, two is the mechanism of disease, and three is rationale of therapy. Think about it. Workings of human body. What does it mean? Human body anatomy, the workings is physiology. So meaning that cell affects both the anatomy and also the physiology of a living organism. The second one is what? Mechanism of disease. What is mechanism of disease? Pathology. And the third one is what? Rationale of therapy. What is rationale of therapy? Pharmacology. So cell talks about the entire medicine, all right? So that's why it's extremely important. Another thing you need to know is that cell has a theory. First of all, the scientific study of cell is known as what? Cytology. There are a lot of professors that studied, cell, which I'm not really much interested in. I'm just interested in the product. Now, I want you to think about cell tenets, something we call the cell theory. You need to know them because in the exam, they may tell you which of the following is not. Which of the following is not? How do you know which one is not when you don't know which one is? So for you to know which one is not, let's discuss which ones are the theory of cell, all right? First, all organisms are composed of cells and cell products. Second, cell is the simplest structure and the functional unit of life. Then number three, an organism's structure and function are due to the activities of their cell. And number four is that cells come from pre-existing cells and not from non-living matter. Number five, cells of all species have many fundamental similarities in their chemical composition and metabolic activity. You need to put it in your head, all right? So that you'll be able to attempt questions on cell theory because they can ask you a whole lot of questions on these five things I just mentioned. Another thing you need to know is that cell has different shapes and you should be able to identify these shapes. There are about 200 types of cells in the human anatomy and millions of cells actually that make up tissues, all right? Now, in terms of cell shapes and sizes, we have the squamous, we have the polygonal, all right? The squamous are thin like pizza. I'll show you a diagram, thin like pizza. You understand? The polygonal, they are irregularly shaped, all right? They have 4 or more sides. You see them, different sides, all right? Then we have the stellate, the one that looks like star. We have the cuboidal, that looks like a cup. We have the columnar. We have, the columnar are just tall, like columns, all right? Then we have the spheroidal, all right? They're round, like the oval office. Then we have the discoid. You understand? It's just like a disc shape, just like the red blood cell. Then we have the fusiform, which are thick in the middle, and fibrous, also, which are thread-like. The fusiform are usually thick in the middle and tapered at the ends. They can ask you a question like this. I know I've seen a question like this, which I will show you at the middle of this discourse, also at the end of this discourse. I want you to look at this diagram, all right? Look at the cell shapes. This is the squamous I mentioned. This is the cuboidal. This is the columnar. This is the polygonal. You see, it has more than 4 sides. Some of them has 5. Some of them has 6 sides. This is the stellate, like star. This is the spheroid, circular. This is the discoid, like a disc. This is the fusiform. You see, it's tapered at the 2 ends. This is tapered at the 2 ends, swollen at the center, like thread. Then this is fibrous, all right? Extremely important for you to know this because a question is going to come out from this. Now, when you are talking about the components of a cell, mind you, the cell is divided, it's housed rather by the plasma membrane. The plasma membrane is like a cloth. You see my clothes? I have the inside and the outside. This outside is known as extracellular. The inside is known as intra. All right. So if you can imagine a room, the wall is the plasma membrane. All right. The wall is the plasma membrane, and the room itself is intracellular. Now there is the inside. This room is known as what? The cytoplasm. That intracellular environment is known as what? The cytoplasm. The cytoplasm is composed of fluid and organelles. All right, extremely important. So organelles in fluid. That fluid is known as what? Intracellular fluid or cytosol. While the outside, usually cells are, the outside of environment of the cell is known as what? Extracellular. And the fluid outside is known as what? Extracellular what? Fluid. Sometimes they can ask you, what are the examples of extracellular fluid? Plasma is an extracellular fluid. It's not inside the cell. Also, the interstitial fluid. You should know what interstitium is. In between two tissues. Remember, cells make up tissues, all right? So cells that are in different tissues, all right, when they want to interchange something and it falls in between those tissues, it becomes what? Interstitial. That fluid is interstitial fluid, all right? Sometimes if protein is, in the interstitium, it can draw fluid into it, leading to edema. But we are not discussing pathology right now. So the basic components of cell is the plasma membrane, all right, the cytoplasm, and what? The nucleus. Or the cytoplasm and what? The nucleus. The cytoplasm is made up of, the fluid, which is the cytosol or intracellular fluid. It has a cytoskeleton that supports it, like pillar, that supports the cell membrane and gives it shape. And also it has what? Organelles, which we will discuss. All right? This is called the cellular trinity. The cellular trinity. They will give you a question. They will tell you every cell has so and so components, but which one is not one of them? All right? We will discuss at the end of I'll give you some more questions at the end of this study. Now let's discuss them one after the other. The first one is the plasma membrane or the cell membrane. As you are looking at this diagram, this is the cell membrane. I need you to understand it with the diagram, all right? This is, the phospholipids. First of all, you need to know that 98% of molecules in the plasma membrane are what? Lipids. 98% are lipids. So there are three types of lipids in the plasma membrane. We have the phospholipids, we have the cholesterol, and we have the glycolipids. So this is the phospholipids, this is the cholesterol, all right? And this is the glycolipids, this one up here. The question is, how do you separate them? First of all, the phospholipid, you see how big the phospholipid is. The phospholipid makes up the phospholipid makes up almost 75% of the lipids. So lipids are 98% of the cell membrane. So among that 98%, 75% is what? Phospholipids. Extremely important that you understand this. Then 20% is what? Cholesterol. So this is cholesterol, this is cholesterol. You see it? Light green. This is cholesterol, this is cholesterol. Extremely important for you to understand. And what's the third one? The third one is about 5%. That's glycolipids. Anything glyco means that carbohydrate moiety is on it. All right. So this thing that is like a 3 is what? Is carbohydrate. All right. So this carbohydrate is on this lipid, meaning that this lipid is what? Glycolipid. Glycolipid. Extremely important that you know this. Now, in terms of protein makes up 2%, of the molecules found in the membrane, but it's heavy. Protein is always heavy. So despite the fact that it's 2%, it's almost 50% of the entire weight. So when we are talking about protein, there are different types of protein. All right. We have transmembrane protein. Like if you look at this transmembrane, it moves from the extracellular to the intracellular. This is transmembrane protein. This is transmembrane protein. All right. It's very, very important for you to know. Transmembrane protein, they pass through membranes. They have a hydrophilic region, which is in contact with the cytoplasm. So, assuming this is the inside of the cell, this is the hydrophilic region. Also, we have the peripheral proteins, all right? This peripheral protein here, all right? Sometimes we have the peripheral protein around here. The peripheral proteins, they are very, very important, all right? Then we have the glycoprotein. The glyco, just like I mentioned in glycolipid, this is a carbohydrate on the lipid, makes it glycolipid. This is a carbohydrate on the protein, makes it glycoprotein. You need to understand. So that when you see the diagram, you should be able to understand it, all right? Now, we just classified these proteins based on their position. We can classify these proteins also based on their functions. Extremely important for you to know this. We can classify them based on their functions. Now, if you want to classify proteins based on their function, how do we classify these proteins? One, we can classify them as receptors. All right? What are the receptors? They are the ones that receive information. They communicate. They are attached to the first messenger. Below them, inside the cell, they are attached to the G protein, which leads to conformational change, all right, in the adenylyl cyclase to the second messenger, all right, to the kinases, and then trigger changes within the cells. That's receptors. Extremely important that you know this. I always call them bouncers. They are always at the door, checking who will enter or who will not enter. Also, we have enzymes, all right? We have enzymes. This one helps to produce second messengers. The second messenger is CAMP inside the cell. Remember that first messenger is outside, second messenger is inside. All right. So the enzymes help to break down chemical messengers and hormones whose job is done. You are done with your job, they break you down. That's the function of the enzymes. Another proteins that are on the cell membrane is the transmembrane protein, or what I usually refer as the channel proteins. Channel proteins are extremely important. All right. If you look, this is a channel protein. As I mentioned initially, this is a channel protein. All right. It's also a transmembrane protein because it moves from here to here. Channel protein. What do these channel proteins do? All right. They allow water and dissolved ions to pass through them. Some are constantly open, the ones that are constantly open. And some are gated, meaning that something has to trigger them for them to open. So they are gated. They need a response to stimuli. That stimuli can be in form of chemicals like ligands, all right? It can be voltage, or it can be mechanically opened. But mind you, they play a very important role in the timing of nerve signals and muscle contractions. Extremely important. Channel proteins, very important to understand this because it is the bulk among the bulk of your questions. Also, we have the cell identity markers. They are usually glycoproteins. All right. This is a glycoprotein. So usually most glycoproteins are cell adhesion, cell identity markers rather, right? What do? They act as identification tag. You want to go to the White House, you must have a tag that shows that you're a visitor. That is what identity markers do. They enable our bodies to identify which cells belong to where. They also enable our cells to identify foreign invaders. This man is not supposed to be here. This lady is not supposed to be here. What we identify them because they are not wearing that tag. They have been identified by the cell identity markers. And these cell identity markers are a type of cell membrane protein. Very important for you to know this. Now, the last cell membrane protein that I want to talk about is the CAMs. That's the adhesion proteins, right? What do? They help to adhere. They help one cell to adhere to another cell. Usually, cell use it to move. A lot of special events, like maybe sperm binding to egg, or binding of immune cell to a cancer cell. It always requires CAMs. We call it CAMs, C-A-M. That's cell adhesion molecule or molecules. And based on the identity markers, what they usually have, remember I told you the identity markers are glycoproteins. What do they have? Carbohydrates. That carbohydrate, we call it glycocalyx. Those glycocalyx are the badge that you are given. All right? They are fuzzy carbohydrate codes that acts like cellular ID badge, name tag. Very important for you to know. Extremely important. So this is this is cell membrane. This is different types of protein. I want you to pause this video and study this very well. When we talk about cellular appendages, which is also another bulk of your question, we talk about three most important things, which is the flagellum, the cilia, and the microvilli. Starting from the last, the microvilli, right? They are just extensions of membranes that are just like 1 to 2 micrometer. You understand? 1 to 2. Very tiny, right? What do? They help to increase cell surface area. Usually you find them in absorptive surfaces like the small intestine, all right? They make absorption more, all right? So some of them are found in very dense areas, in fridge compartments. We call them brush border. Extremely important for you to know about microvilli. Extremely important. Another one, you talk about cilia. Cilia, they are like hair processes. These ones are bigger than microvilli because they are like 10 to 10 micrometer long, all right? Some of them are motile, some of them are non-motile. They are found in different places, all right? The ones that are non-motile, that's the primary cilia, they are usually found in sensory areas, like sensory inner ear, all right? Like the retina, the nasal cavity, the kidneys. Those are the places you see the non-motile cilia, all right? But the motile ones are usually found on respiratory surfaces, even the uterine tubes. It helps to move the egg, to prepare the egg. That's motile cilia, all right? Even in the brain, the ventricles of the brain, you see cilia there. All right. Even in the male, you find it in the ductus. That's why if there's a problem, like a child that has cystic fibrosis has a problem in the cilia. Yes. Because the cilia has somewhere they call the saline area. It has the axoneme and the saline layer. If there's a problem in that saline layer, right? That saline layer is the chloride pump. It pumps chlorine into the ECF, pumps chlorine out. So if he has a problem, there's accumulation of chlorine. And before you know it, that child is always having respiratory problem and that child will be infertile. Why? Because cilia is found help to propel the egg forward. It's found in the uterine tubes. And also in male, cilia is also found in the testicles, the ductus of the testicles. Very important for you to know this. Also, we have the flagellum. The flagellum is the biggest, it's a long wipe that, power the sperm movement. So it's extremely important. So we have the flagella, we have the cilia, we have the microvilli. So you need to know what is found where. What is found where. Now let's talk about the membrane layer, all right? Because there is selective permeability of that layer. Remember, the phospholipids, right? The two heads of the phospholipids are in and out. They are hydrophilic. The tails are inside, which is what if you look at this, these are the tails. The tail is hydrophobic, while the heads here and here are hydrophilic. So they help to curtail what goes in and what doesn't go in. Like smaller non-polar molecules can easily go in, like oxygen, CO2, N2. You understand? But the ones that are a little charged, they all might not go through. All right. Some might go, some will not go, like glycerol. All right. Things like glucose, sucrose, they need carriers to move in. Ions cannot enter inside or go out. Can't go out, except maybe they are gated. There is a change, in the stimuli that leads to them going out. So this leads us to major transports. We have the plasma membrane transport, right? Because it's selective permeable. We have the passive transport mechanism, which requires no ATP, and we have the active transport mechanism, which consumes ATP. Also, we have the carrier-mediated mechanism, passive, active, carrier-mediated mechanism. Example of passive: filtration, diffusion, osmosis. Example of active transport, vesicular transport. Example of carrier, we will discuss that later. So you have to bear in mind that filtration uses hydrostatic pressure. That's what they can ask you in filtration. All right. Know about the simple diffusion, just movements of like the net movement of particles from one area of higher concentration to an area of lower concentration. All right. Usually because of their motion, constant motion. That's why it's called what? Simple. It requires what? Concentration gradients, right? Now, you also need to know factors that affect simple diffusion, like temperature, weight, steepness of concentration, membrane surface, membrane permeability. Extremely important for you to know. Now, osmosis is talking about water. That's the flow of water from the side of a selectively permeable membrane to another side. Extremely important for you to know. And also, I need you to understand that there are something we call aquaporins. Aquaporins are channel proteins, all right, in the plasma membrane that are specialized for passage of what? Water. So the cells use it to increase the rate of water coming in or out, or reduce. That's aquaporins. Aquaporins. All right. Also, I need you to remember that osmotic pressure, osmosis is the movement of water. We already discussed that. All right. Osmotic pressure leads to osmosis. That's really important. And you need to know the difference between osmolality, L-I-T-Y, and osmolarity, R-I-T-Y. You need to know this. When you're talking about riti, that's R. You understand? You're talking about the number of osmoles of solute per liter of a solution. So it's talking about volume. But osmolality is talking about weight. So usually in kg. But this one is in liters. Extremely important for you to know. So usually 1 osmole is equivalent to 1 mole dissolved particle. All right? So you look that up. Very important. I'll give you a question based on that. Now, we also talk about tonicity, all right? Tonicity is the ability of a solution to affect fluid volume and pressure in a cell. It always depends on the concentration and permeability of what? Solutes. We are going to discuss that because that will lead us to talking about the hypertonic, hypotonic, and isotonic solution. I'll shed a little light on it because it will come out in your exam, and I'll give you a question on that, all right? When you're talking about hypotonic, mind you, always consider the external environment, not the internal environment. So this is the solution. Something is inside. That solution outside is hypotonic, meaning that it has less concentration of what? Solutes and more concentration of water. Simple. So what will happen to that cell inside that solution? The cell, meaning that the cell has higher concentration of salt. So that cell will what? Draw water into it and swell and burst. So a hypotonic solution will lead a cell that is in that hypotonic solution to draw water. That's how you think it. When you talk about hypertonic, think about that a bucket of water, all right, and you put something on it. That bucket of water that has a lot of salt or sugar in it, that's solute. High concentration of solute is what? Hypertonic. Now that thing you put inside it is what? Cell. So If it's hypertonic, it means that the cell, the solute in that container is more than the solute in the cell. So what will happen? That container will draw the contents of the fluid content of that cell out, leading to shrinking. So when you're talking about hypotonic, you're talking about low concentration of solute in that solution. Hypertonic, high concentration of solute in that solution. Isotonic, equal concentration. Simple. If you put it that way in your head, because a lot of students, when they hear hypotonic or hypertonic, they're thinking, oh, we are talking about the cell itself. No, we are talking about the surrounding where the cell is in. So they're comparing that surrounding with the content of the cell. Simple. So usually the question is about whether the cell is going to swell or shrink or remain the same. So you need to think about it that way. Now, I'm not going to talk much on carrier-mediated transport because you have uniports that they carry only one solute at a time. We have symports. You know what symport is? They carry two molecules. You understand? Simultaneously. That's co-transport. Another name for symport is co-transport. Then we have what? Antiports. They carry two different molecules in opposite direction, like the sodium-potassium pump, all right? Sodium-potassium pump, which brings in potassium and removes what? Sodium from the cell. We know this, all right? So we also have some methods that uses carrier, like facilitated diffusion, like active transport. They use carrier as well. You understand? They use carrier as well, but does not consume ATP. So diffusion does not consume ATP, whether it's carrier-mediated or not, all right? Extremely important for you to know this. But active transport uses what? ATP, like the sodium-potassium pump. They use ATP. The carrier-mediated and their active transport. I want you to bear this in mind, all right? Now, the one I also need you to focus on is vesicular transport. Vesicular transport, which is, usually the processes that move large particles, fluid droplets or nutrients, at once through the cell membrane in vesicles. Vesicles means bubbles. That's vesicular transport. Example is endocytosis and what? Exocytosis. Endo and exo. Endo and exo. Does it make sense? So when you are talking about endo, there are three types of endo. We have phagocytosis, we have pinocytosis, and we have what? Receptor-mediated endocytosis. Very, very important for you to know. Phagocytosis, cell eating. Endocytosis, cell drinking fluid. Receptor-mediated, they use what? Receptor. This is the receptor. This is a receptor that attaches to whatever it wants to take in. All right? So this is the method. This is called what? Vesicular transport. Endocytosis. Now we have also what? Exocytosis. So we have exocytosis, which is when the cell packages those things in vesicle and push them out of the cell. Very straightforward. Now, looking at the internal environment of the cell, the first thing you want to think about is the cytoskeleton. All right? What does the cytoskeleton contain? That's the kind of question that we ask you. Talk about microfilaments, all right, which are the actin fibers of cell that helps for cell movement and shape. We have to talk about the microtubules. You understand? They are like highways, transport highways. We talk about the intermediate filaments, very, very important for strength. And we have also talk about the terminal web, right? These are network that connects every entire thing in the cytoskeleton. These are the four major components of the cytoskeleton because they will ask you, which one is not part of the cytoskeleton composition? Do you understand? Like that is telling you must know others. So the cytoskeleton is a cellular construction crew, right? It's a crew that helps in, just like people that construct the road or bridge. That is what the cytoskeleton does. So it has all these things. Sometimes there can be cilia, but it's just more like an accessory. That's why we don't put it as part of the cytoskeleton. All right. Now, this is the rough diagram showing the internal environment of the cell. We have this membrane, the nucleus, the nucleolus, lysosome, cytoplasm, ribosome, Golgi, vacuole, mitochondria. You need to know all these things, all right? And you need to know the membranous organelles and which ones are not the membranous organelles. You need to know what inclusion is, all right? When you talk about membranous, you are talking about the VIPs, the bourgeoisies, the ones that have the best of all clothes, all right? Extremely important. Examples like the nucleus, the mitochondria, the lysosome, the perisosome, the endoplasmic reticulum, the Golgi. These are the ones that are membranous. They have clothes. They are the VIPs. But the ones that are not membranous, you talk about the ribosomes, the centrosomes, the centrioles. So you need to know any of the two groups so that you'll be able to answer that question in the exam. Because they may ask you, which is not a membranous organelle? What do you tell them? Ribosome or centrosome or centriole? Also, we have what we call inclusions. Inclusions, all right? Inclusions like fat droplets, like dust, all these things, the things that are not even supposed to be there. They are never in a membrane, never. They are also non-membranous, all right? So extremely important for you to know. So my assignment for you is go and read up the functions of these organelles, especially the nucleus, which we know, it contains the most important thing. Inside is the nucleoplasm. It contains the chromatin, and the chromatin is the DNA and the protein, right? Yes. Then we have the nucleoli, which are more like where ribosomes are being produced. All right, very, very important for you to know. Then we have the endoplasmic reticulum, which is divided into two. We have the one that looks rough because it has ribosome, it produces proteins, and the one that looks smooth because it does not produce what? Protein. It produces lipid. It does not have like ribosome, so it produces lipid and synthesizes lipid rather. Synthesis. All right. Very, very important for you to know this as well. Also, the ribosomes, we discussed that more in genetics, right? They are granules of protein and RNA. Very, very important for cell activities. All right. Then we have the Golgi complex. If you've worked in Amazon before, you understand what I'm saying. They help to package. So they take things, they package it in Golgi vesicles. All right. Extremely important. They also help to synthesize other things like what? Like carbohydrates. They add carbohydrate moiety in that packaging. Extremely important for you to know the Golgi. It's always green, as you can see here. All right. Then we have the lysosomes as well. All right. These lysosomes, they help in they have enzymes. They have enzymes, so they help in lysine. Their function is usually what? Intracellular hydrolytic digestion of proteins, nucleic acids, and other things. They also undergo autophagy and autolysis. Let me show you the lysosome. The lysosome. You see? Yes, this is lysosome. You see? This lysosome. Extremely important for you to know what the lysosome is. You understand? Because it really helps in autophagy. Something we call autophagy. That's digestion and disposal, of other organelles that are old. Also the autolysis, cell suicide. So that's the lysosome. Then we have the perisosomes. They resemble the lysosome, but they contain different enzymes, and they are not produced by the Golgi complex, because the Golgi complex is what produces the lysosome. All right. Instead, the perisosome, they contain all these hydroperoxidase, catalase, all these things that helps to break free radicals. Also very important for you to know that. All right. The mitochondria is the powerhouse. Everybody knows this. Mitochondria is the powerhouse. What am I forgetting? My inclusions. Okay, I've already discussed inclusions. So these are extremely important. This is cell. Honestly, we just broke down cells. So it's all about you listening to this video many times for you to be able to get it. All right. So there are some questions that you may be meeting. Very straightforward questions. Like what? Like, they can ask you which of the following is, let me see, which of the following statements is not true of modern cell theory? And we already discussed that. Modern day theory. So they will give you all the different modern day theory, and they will, they will forget, they will give you one that is wrong. For example, like all cells occupy space. You understand? So the truth of the matter is that all cells occupy space. That is true, but it's not the fundamental principle. You see how they can confuse you with this kind of question. So you need to be very, very wary, all right? Also, they can ask you that about cells, the shapes of cells. They say cells that are thick in the middle and taper toward the end are called what? I want you to put the answer in the comment section. If you look at this, what do you see? What do we say about fusiform? I mentioned here that fusiform are thick in the middle and tapered at what? At the end. Sometimes they can ask you which cells are flat and are usually found in protective places. Example, skin. You tell them squamous. Sometimes they may not even tell you skin. They may tell you esophagus, squamous. Sometimes they may tell you the anus, squamous. The vagina, squamous. So you need to know that flat cells are usually what? Squamous, meaning that you need to know the shapes. Sometimes they can ask you about the cubes. Sometimes they can ask you about the polygonal. So you need to be able to get the question, from the option. Get the answer of the question from the option. Sometimes they can ask you about the fluids. They can say the fluid inside of the cell is called what? Cytosol. They can tell you cytosol, they can tell you intracellular fluid. They can also ask you the fluid outside of the cell. What is the name? These are things you need to know, all right? Sometimes they can even tell you, which of the following is not one of the three basic components of a cell. Do you remember the components of the cell that I mentioned initially? What are they? Talk about the nucleus, talk about the plasma, talk about the cytoplasm, all right? They can give you the other components of the cell. So they will give you one that is not a component of the cell. They can tell you interstitial fluid. They can tell you extracellular fluid. Are they components of the cell? No. They can ask you a question. They say, a question like on transmembrane. They can tell you most transmembrane proteins have dash region facing the intra and extracellular fluid and dash region embedded in the phospholipid. What did I tell you? I told you that this is the phospholipid, right? The internal part of this phospholipid is what? Hydrophobic. While this external part, up and down, is what? Hydrophilic. Simple. Also, they can ask you the most important molecules in the plasma membrane are dash. And they give you options like glycolipids, phospholipids, prostaglandins, proteins, cholesterol molecules. What is the answer? It's as simple as phospholipids. Why? Because they make up 98% of the plasma membrane. And this question is asking you about the plasma membrane. It can't be glycolipids. It has to be phospholipids. Because glycolipids is just 5% of that 98%. But phospholipids is about 70-something%. It's not prostaglandin. We never talked about prostaglandin. It's not even proteins, because proteins are just 2%. It's not even cholesterol. Cholesterol is around 20. So the answer is what? Phospholipids, if asked. Now, they can ask you about the jobs of the protein, like asking you about the proteins that do not protrude, all right, into the phospholipid bilayer, but they are the surface. What are they? Surface, that's peripheral, all right? You need to pay attention to this. Sometimes they can ask you about the glycoproteins. You understand? They can tell you which one wears the sugar coat on his head. A protein that is bound to carbohydrates. What is it? Glycoprotein. I already discussed this. Sometimes they can ask you the plasma membrane are made up of what? A molecule that has, a hydrophilic head and a hydrophobic tail. What is it? Phospholipids. So most questions are surrounding that, surrounding the plasma membrane. Sometimes they can ask you about the channel receptors, like asking you a question about the membrane proteins that bind to the specific membrane protein that binds to signals by which cells communicates. It's called what? Receptors. All right? Because the first messenger attaches to the receptor, and then leading to a conformational change inside through the adenylyl cyclase, the CAMP, which is the second messenger, and the kinase and the enzymes. All right? So it's very, very important that you know this. And sometimes they can also tell you that a receptor protein in the plasma membrane will not bind to just any chemical in the extracellular fluid, which is true. Why? Because these are enzymes. These are protein enzymes. And protein enzymes are very specific. So due to their specificity, that is why a receptor protein in the plasma membrane will not bind to just any chemical in the ECF. It has to be specific to the one it is programmed for. That's specificity. All right. Oh, before I forget, they can ask you a question on the first and second messenger. All right. For example, this question, they tell you that the CAMP, that's the second messenger, activates enzymes called dash. What enzymes does the second messenger activate? If you remember the pattern or the pathway I discussed during the initial part of this particular video, I mentioned that the first messenger attaches to the receptors, leading to a conformational change inside the cell. So inside the cell, because that receptor is a transmembrane protein, it's connected inside and outside and inside. So once this one is connected outside, that's the first messenger, down on its leg, you see the G protein. That G protein will get activated and activate what? Adenylyl cyclase, which is also at the membrane. The adenylyl cyclase will now activate ATP. ATP will now go and activate CAMP. That CAMP is the second messenger. Now the CAMP will now activate kinases. So the answer should be kinases. It is the kinases that will now helps to, shut off some enzymes or activate some of the enzymes inside. So I need you to understand this. You need to know this sequence so that you don't fail this question. Okay, looking at the glycocalyx, for example, in this diagram, what do you see? The glycocalyx are actually glycoproteins, all right? If they are on proteins, they become glycoproteins. And what does these proteins do? I mentioned to you that there are some proteins that helps for what? Identification tag. They are identification tag. So anytime you hear identification tag, they are glycoproteins. They are glycoproteins, which is when glycocalyx, that's carbohydrate, is perching on a protein. We also have glycolipids, but the answer should be glycoprotein. So anything about identification, glycoproteins, glycocalyx. Glycoproteins, when it is on glycocalyx, when it's on protein, it becomes what? Glycoprotein. Always remember this, identification tag and also identification. Now, there's a question that asks about appendages. For example, which of these appendages provide a motility to a sperm cell? Answer. Pause it and give the answer. Flagella. Dash acts as a sensory antenna in many cells. What acts as a sensory antenna in many cells? Cilia. Which one increases cell surface? Microvilli. You need to know where each and every one of these things are found. Like microvilli is usually in absorptive surfaces like the intestine. All right. Then we have the cilia, which we have the mobile and the non-mobile. Know where the mobile is found, know where the non-mobile is found. But this is the best way they are going to call in this question. Also, they can ask you, for example, that's exactly why I was mentioning initially in the last few minutes ago, that you should know which one is found where. For example, they can ask you, which one of the following is found in the intestine, or one of the following is used for is specialized for absorption of nutrients. What is the answer for the two of them? Microvilli. So when a membrane allows some things to pass through it, but not others, what is it called? Selective permeability. Remember, selective what? Permeability, which I discussed initially. All right. Another question they are going to ask you is about that's in terms of permeability of the cell is what can get in and what cannot get in. That's why this diagram is there. What can get in and what is it permeable to? It's permeable to what? Nutrients. Nutrients we get in well, comfortably. It has channels for it. It has everything for nutrients to get in. But what cannot get in? Proteins cannot get in, mind you. So nutrients can get in, proteins cannot get in. So it's permeable to nutrients, impermeable to what? Proteins. Extremely important. This question is very technical. Now, they can ask you a question on filtration and hydrostatic pressure, all right? When they ask you anything about forces of water, how water moves, that is what? Filtration. Like they want to filter. Dash is the process in which dash forces water and small solutes such as salts through narrow cells between capillaries. You are talking about filtration. Filtration, you need to know the difference between filtration and osmosis. Osmosis is just movement, right? But when hydrostatic force is there, through between capillaries, you're talking about filtration. So filtration and hydrostatic pressure, not osmosis and hydrostatic pressure. Mind you, you need to understand this. That's why I need you to pause and zoom these PowerPoints and understand what I'm talking about. And also, when they are telling you that about water moving from through selectively permeable membrane is in a process called osmosis. We all know that, all right? But when it's talking about water crossing channels of transmembrane proteins, what is it called? Aquaporins. When it talks about channels, water crossing channels, this is aquaporins. But semi-permeable membrane, normal movement, that is osmosis. In terms of moving into the capillaries, that is filtration. Using what? Hydrostatic pressure. Now, they can ask you a very clinical question. They may use insulin, they can use anything, all right? And they tell you that, insulin moves from, the endothelial cells that line the blood capillaries and move into the cell, across the cell. What is it called? It is transcytosis. Trans movement. Trans from here to here. Transcytosis. All right? Extremely important because they'll give you options. They'll give you like pinocytosis, that's cell drinking, receptor-mediated phagocytosis, passive transport, facilitated transport, this blah, 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 this and that. But know that we are talking about what? Transcytosis. Also, they can tell you about sodium and glucose when they are transported together. Sodium and glucose when they are transported together. All right? The carrier protein is a symport. And, they can also ask you the process. The process is co-transport, all right? Co-transport, moving together. Very, very important for you to know. So symport, co-transport, because they can try to confuse you in different ways. Now, they can ask you about sodium and potassium pump, which we mentioned, all right? Sodium and potassium pump. Remember, they are moving sodium and potassium pump moving up their concentration gradients, up. And this is a process of what? Active transport. I mentioned active transport. Extremely important for you to know. Active transport. And sometimes they can ask you, what does facilitated diffusion and active transport have in common? Both of them are carrier mediated. I mentioned it earlier. Both of them are what? Carrier mediated. What do facilitated diffusion and active transport have in common? So whenever they ask you anything about energy in terms of this movement, is always active transport. They can give you options like filtration, osmosis, simple diffusion, facilitated diffusion, this, that. Answer is always active transport as far as ATP is involved. That's a very important information for you to know. And when they're talking about when particles moving inside the cell, that's endocytosis. Endocytosis, remember I told you that they are divided into three, which is pinocytosis, phagocytosis, and what's carrier-mediated. Pinocytosis is what? Cell drinking. You need to know what they're asking you. So which of the following is not a method by which particles can live? And they give you something like an antipod system. Yes, particles can live, of course. Simple diffusion, of course, particles can live. Exocytosis, yes, particles can live. Active transport, yes, particles can live. But if they give you something like a phagocytosis, that should be the answer. Or pinocytosis, that must be the answer. I'm stressing on this because they asked a lot of questions about this. That's why I'm stressing on. You understand? So phagocytosis is cell eating. You understand? Like, for example, if there is a bacteria in the system, white blood cell engulfs it, uses its lysosome to kill it. That bacteria becomes a phagosome. So it forms a complex known as phagolysosome. After destroying it, it pushes it out through what? Exocytosis. So when blood cells engulf bacteria, it is what? A method of phagocytosis, cell eating, not pinocytosis. It's phago. But if it is fluid, it's known as what? Pinocytosis or pinocytosis. Anyone that works for you. Now, they can ask you something about what is the function of sodium and potassium pump. You need to know what the function is. Remember, it's an antipop type, right? An antipod, not a sympod. Antipod type of co-transport, all right? So what does he do? He helps in production of heat, of course. Secondary active transport, yes. Regulation of cell volume, yes. Because those movements, water follows those movements. Maintenance of membrane potential, of course. But look at this one. Transport of sodium into sodium has to be transported out. You have to be aware, because most times when you just see it, you know it's potassium and sodium and potassium transport, all right, going in the opposite direction. You'll be like, okay, that should be the answer. No, which one is not? Has to be this, because sodium is being pumped out. Potassium is pumped in. So if I ask you if you are asked an example of active transport, you should be able to know. All right. Or any question on pinocytosis, you should be able to get the answer. All right. Also, I need you to know what affects the rate of diffusion. I said it initially, I'm going to repeat it. Size of molecules, steepness of concentration gradient, temperature, membrane permeability. Of course, all these things determines, the rate of diffusion. All right. How about enzymes? Does it determine? No, has nothing to do with enzymes. All right. Remember this. Now, they can ask you a question on tonicity. All right, about hypertonic, hypotonic, and isotonic. I've already discussed it, so I'm not wasting time on it. You can say a question like, which of the following contains higher than normal amount of osmotically active solutes? You should know that's hypertonic. If they say lower than normal, that's hypo. And they can ask you, normal, that's, iso. What if they tell you that a red blood cell is placed in a hypertonic solution, right? What will happen to that red blood cell? This means the concentration of solutes in the solution is what? High. So what will it do? It will drag out the water from the red blood cell, and the red blood cell will what? Shrink. So higher concentration of solutes and shrinking. And what if they ask you that red blood cell put in a hypotonic solution? What will happen to it? Well, first of all, what's the amount of concentration of solutes in that solution? Less than the one inside the red blood cell. And what will happen to that red blood cell? Red blood cell, because it has higher salt and less water, it will attract water and swell. All right, swell. So you need to know what happens to which. Also, remember osmolarity and osmolality and tonicity. Tonicity. We know tonicity. Tonicity is just the ability of a solution to affect the fluid volume, right? That's tonicity. It's as simple as that. And so, and when we are talking about osmolarity, it's the number of particles of a solute in a solution. So it's R-rity. Sometimes they may be asking you about the cytoskeleton. They will tell you, what helps gives the cell the structural support, determine the shape of the cell, and also directs the activities of blah 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 blah. All is you immediately hear support the cell. You are talking about cytoskeleton. It's just the skeleton of the cell. All right, it's just a skeleton. The plasma membrane is just like the flesh. So the cytoskeleton supports the flesh. All right. If you just hear that, you don't even need to think twice. All right. And also you need to know what the cytoskeleton contains. Told you, microfilaments, microtubules, terminal web, intermediate filaments. Now they can give you options and say which of the following is a membranous organelle or which one is not a membranous organelle. So you should know which ones are and which ones are not. I won't dwell on this more. Also, they can ask you the functions of cells, of organelles rather. For example, they say among other functions, hepatocytes, that's liver cells, are specialized for detoxifying drugs or other chemicals. Hepatocytes have large amounts of dash. Think about it. What is helping in detoxifying? It's smooth endoplasmic reticulum. If you look at the functions, one of its functions is to, for detox. Very important. I don't know if I mentioned this in my discourse, but this is a very important information. Now, what is the function of the ribosome? I mentioned it. Protein synthesis. That's why, like the rough endoplasmic reticulum helps to synthesize proteins because they contain what? Ribosomes. So if there's anything that happens and somebody's ribosome is not functioning very well, right? So a question, for example, what would immediately cease if the ribosome of a cell were destroyed? That's synthesis of protein. It's as simple as that. Just synthesis of protein. And also, if you look at the lysosome, a very, very important organelle because they ask a lot of questions about it. For example, which organelle is most active in causing programmed cell death? That's lysosome. Remember what I mentioned? When organelles are worn out, it destroys them. That's the function of the lysosome. And it's produced by what? The Golgi, usually. And when you are running muscle cells, what does muscle cell contain that gives you a lot of ATP? Mitochondria. These are the way they can ask you a lot of things on these cells. They can also tell you which organelle synthesizes steroids. Remember what I told you about lipids? What synthesizes it? Smooth endoplasmic reticulum. That's steroids. And which one helps in cell division? Centrioles, microtubules. Those plays an important role in cell division. And that's centrioles. And centrioles are made up of what? Microtubules. What if they ask you what synthesizes carbohydrate? I mentioned it initially. And also, helps to put finishing touches on protein. That's packaging of protein. That's the Golgi. They want to confuse you. What if they ask you about which of the following decomposes fatty acids, leading to detoxification of alcohol and free radical? Immediately you hear free radical, think about what? Perizosome. If perizosome is down as an option, pick it. And also they will ask you which organelles are involved in protein synthesis. Rough ER, Golgi, nucleus, ribosome. These are the 4 organelles that are specifically involved in protein synthesis. You see these organelles? Extremely important. And that's what they will give you an option. So look for the one that is not part of these 4 and pick as the answer. Sometimes they can also ask you about the organelles that sorts, cuts, splices, packages. If you hear packages, Amazon, Golgi. All right? Sometimes they can ask you which organelle, is double membrane organelle consisting of a matrix and crystal. This one is mitochondria. I didn't mention it. And thank God I see something similar to it now. So know that the mitochondria is the powerhouse of the cell, and also it's an organelle that is double membraned, all right? And consists of what? Matrix and crystal. Extremely. Go and look at the diagram of endoplasmic reticulum, of mitochondria rather. Go and look at the diagram properly. You will understand what I'm talking about. Let me see if I can see a diagram. Look at it. Look at the crystal. You see? Look at the membrane. You understand? So you have a double membrane, this and this. And also it has a matrix and it has a what? A crystal. Extremely important for you to know. And this is the Golgi. Helps to package, helps to synthesize carb. Very, very important. They will also ask you about inclusion. Inclusions. What are inclusions? Inclusions are things that are not really, they are things that the cell can do without. Example, fat droplets, glycogen granules, bacteria, dust particles, all these things, when they find themselves inside the cell can do without them. They are called what? Inclusions. They're just included. So you need to also understand this, all right? And they are never enclosed in a cellular unit. So the last question I'm going to ask you is, what are accumulated cell products that are never enclosed in a unit called, all right? What are they called? I want you to put the answer in the comment section, all right? So I just discussed more than 40 to 50 questions right now, honestly, on cells. And I need you to go through these things. Use your pen and paper to write it down and understand exactly what we are talking about. So thank you very much for staying till the end of this video. I hope you learned a lot. If you have any question, if you have something that you want me to discuss, put it in the comment section and I will respond to you very fast, all right? Thank you so much. Make sure you comment. Make sure you like. Please leave a like for me and leave a comment, all right? It's really will help go a long way to encourage me to make more videos for you, all right? Thank you so much. Have a nice one.